and welcome to this video covering the most common styles of exam questions within the cells topic. Now I do find that in this area there tend to be a lot of recall heavy questions, so if you're looking for more examples of those you can look back to the worksheets that accompany all of the previous cells videos, but in this exam styles question video I've particularly focused on those that are the most challenging that we can work through together. So our first question is a cell structure question, and it's a structure function question. So we need to really clearly link structures of a particular cell to their function. And in this case, we are talking about epithelial cells in the small intestine and their role in absorption. And if we have a think about an epithelial cell in the small intestine, the first thing that probably jumps out at you are the microvilli, so the folds in the cell surface membrane. And they are really important for increasing the surface area of the cell, and that would get you 1 mark. But it's not going to get you the 2 full marks. To get your 2 marks for that point, you need to explain in a bit more detail. So what is that surface area for? So you could talk about channel proteins or carrier proteins or what they're involved in, so facilitated diffusion, active transport, but you need to go beyond just saying that it's got a higher surface area. Second marking point is to saying that there are lots of mitochondria packed inside this little cell. And again, what are those mitochondria for? And we need to stress that there are lots of them because loads of other kind of cells, they all have mitochondria, but this one in particular has extra mitochondria because it's doing lots of active processes, and particularly it's active transport that requires ATP. And I've written a really wordy explanation here, but it is important if you're writing about ATP or respiration, you need to be including lots of detail. So the mitochondria are there to make ATP in aerobic respiration, and that's to release energy for active transport. So lots of explanation there, but it is important. And then the third marking points, the sort of pair of marking points, are for talking about things like carrier proteins for active transport, or for talking about channel proteins or carrier proteins for the process of facilitated diffusion. You didn't have to write both of those things as different options, different ways to get your marking points. But again, you can see the linked marking points. So lots of your particular proteins for your particular process gets you the 2 marks. And then finally, membrane-bound digestive enzymes. So digestive enzymes that are found actually on the cell surface membrane, and they are there to maintain a concentration gradient. So they digest larger molecules into smaller molecules on the membrane itself, and that helps to maintain a concentration gradient for absorption of those smaller molecules. The second question asks you to contrast the structure of a plant cell with that of a prokaryotic cell, and essentially this is just asking you for the differences between eukaryotic cells and prokaryotic cells, but it has specifically stated that it's a plant cell, so you do need to be careful with your answer. So firstly, all eukaryotic cells, one of the defining features is that they have membrane-bound organelles, and it is always good practice to include an example, such as, for example, mitochondria. The ribosomes are larger in plant cells. That's an example of a eukaryotic cell. They are 80S compared to the 70S ribosomes in prokaryotes. The cells themselves are larger in eukaryotes, such as a plant cell. And plant cells have a cellulose cell wall, whereas prokaryotic cells have a cell wall composed of murine, which is a form of glycoprotein. You can use the word peptidoglycan, but it's not in the syllabus, so I would definitely go for murine and glycoprotein. And the DNA in a plant cell is linear DNA, so it's linear chromosomes contained within a nucleus. And the comparison here, or the contrast, is that your prokaryotic cell has circular DNA found free within the cytoplasm, so it doesn't have a nucleus at all. And then plant cells have DNA associated with histones, so in the chromosomes the DNA are wrapped around histones, the little proteins, whereas that doesn't happen in prokaryotic cells. There are plasmids within prokaryotes, but not within plant cells. And again, using this table, just make sure you make those direct comparisons, and it forces you to say things, for example, there are no plasmids in a plant cell. And that's the kind of thing that if you didn't write a table, you might be tempted not to write that because it's just the fact there's nothing there. So lots of detail, and a table's a good idea. So question number 3 asks you to produce a scientific drawing of an onion cell. And in this case, they've asked you just to draw the one cell that they've highlighted, and you do not need to include magnification, although that is something you should try and include, unless, like this, you've specifically been told not to include it. So in terms of scientific drawings or biological drawings, there are a few things you need to be careful about. So you need to be thinking about drawing with nice clean lines. you need to draw exactly what you see in the diagram. So in this case, don't start drawing a sort of stylized onion cell, what you think it should look like. Just draw exactly what you see in the image and then label anything that is visible in the image. So if I have a go at drawing this, it doesn't need to be absolutely perfect, exactly the same shape, but you need to make an effort to make it roughly the same shape as you can see on the diagram. So I haven't just gone for a sort of rectangle, I've tried to show you the shape that is in the image. And then I do a double line to represent the cell wall. And then the only other structure I can really see on here is the nucleus, so I draw the nucleus roughly in the same position that I can see it on the diagram. And in terms of marking points, I need to think about labelling as well. So I'll label up my cell wall. And the inner line should be my cell membrane. And then I've also got a nucleus and the cytoplasm. And in terms of marking points here, we need to be thinking about nice clean lines. So not having any sort of crossing over points or hanging lines, no shading. And then you also need to be thinking about labelling up all your labels. And the other mark comes from it looking the same as the image that you're given. So those are my 3 marking points for that question. And the second part of this question is a magnification question, and you are given here your actual and your image sizes, but do be aware in a real exam question you could also be asked to get your ruler out and to start measuring the image size yourself. But we're given both values. Now the first step for any magnification question is to write down your equation, and I would always start with my magic magnification triangle, and I remember it as I am, and that tells me all the equations that I need to know for magnification. So in this case we're asked for the magnification, so M equals I over A, so magnification equals image size over actual size. But you could also work out, so actual size would be image over magnification, and image size would be actual times magnification. Now the second job here is to make sure that all our units are converted into the same units before we start. So we have some micrometers, but we also have some centimetres, and you should never ever use centimetres. We need to get straight out of centimetres into millimetres. So we'd always use metres, millimetres and micrometers. If you start trying to convert straight from centimetres into micrometers, you're more likely to make a mistake. So 2.7 centimetres is equal to 27 millimetres. I'd always do that first rather than trying to do that direct conversion. And then 27 millimetres, because there are 1,000 micrometers in a millimetre, so that is equal to 27,000 micrometers. And then we just do our equation. So magnification equals image size over actual, so 27,000 micrometers divided by 180 micrometers, and that gives you an answer of times 150. You get 2 marks for a correct answer, but always remember that if you do include the correct working, so particularly if you've included your equation and you've shown the examiner exactly what you're trying to do, you can usually get 1 mark for showing the correct working. So do always include all of your working rather than just the correct answer. Question number 4 is another magnification question, and in this case we're given an image of an amoeba with the actual size missing off the scale bar. And we're given quite a lot of information in the question, so your job is to pick out the most relevant bits of information to use in your calculation. Now I would always start with your magic magnification triangle, so I am, and in this case we're asked to calculate the actual size, so actual size is image size divided by magnification. And you're given the image length in millimetres. So your first job would be to convert those millimetres into micrometers, remembering that there are 1,000 micrometers in 1 millimetre. And then you just do your calculation. So actual length would be equal to 41,000 divided by the magnification, which is 150. And that gives you an answer of 273.3 recurring. But we are specifically asked to give our answer into 2 significant figures. So you would give your final answer as just 270 micrometers. Okay, so key things to remember here are don't forget to show your full working, and remember to include just 2 significant figures and to include micrometers in your final answer. Second part of this question asks you to explain why it is not possible to observe ribosomes when you're using an optical microscope or a light microscope. And the key thing here is that the resolution is too low, and it's too low to observe really small structures such as ribosomes, whereas you can observe structures such as your nucleus in the centre of the cell. And the reason the resolution is so low is because the wavelength of light is very long compared to, for example, the wavelength of electrons when you're using an electron microscope. And because of that large wavelength, you aren't able to distinguish really small things such as ribosomes as separate to each other, whereas you can see large objects such as your nucleus. And then the second part of this question asks you to explain how the amoeba is able to rely on simple diffusion to get oxygen into the cell. This is a synoptic question, so it's drawing on different content apart from just the cells topic, so it's drawing on the surface area to volume ratio content. And here you need to be saying that it is a small organism or a single-celled organism, and that gives it a large surface area to volume ratio. So it has a much larger surface area to volume ratio compared to a bigger multicellular organism. And then that means it has a short diffusion pathway. So there's not far for that oxygen to go to get to the middle of the cell. And that means that diffusion is sufficiently fast to meet all of the requirements of the cell. Our final question relates to cell fractionation, which is a really common question within the methods of studying cells topic. And the first part asks you to explain a couple of bits of the student's method. So what he's done is he's taken some spinach leaf cells, he's blended them up with an isotonic solution, filtered his solution, and then used a centrifuge. And you're asked to explain why he used a blender and why he filtered his solution. And in terms of using a blender, that is to break open the cells. So we're looking for the organelles inside, and we need to expose those. So we need those organelles to be free from the cell membrane and the cell wall. So using a blender or some sort of grinding up in a pestle and mortar would enable those organelles to break free from the cell. And then in terms of filtering, we need to remove any large cell debris. So any large bits of cell that haven't broken up completely, or any cells that are just not broken up at all, need to be removed before you carry on and start using the centrifuge to start isolating your organelles. And then the second part asks you about the properties of the solution that he would use. So why isotonic, and then what are the other important properties? And this is a really, really common question within the cell fractionation topic, so it is important that you know this really well. I've used a table, and a table here just really helps you to make sure that each property you've given a specific explanation as to why it's important. So if we start with isotonic, isotonic means the same water potential as the cells, and that's important because it prevents osmotic action and prevents your organelles from bursting, prevents lysis of your organelles, and that's really important because we want a chloroplast at the end. We don't want mush from our burst chloroplasts. And it's important there that you do write about lysis of organelles, not cell lysis, because we're actually wanting to break open our cells. And then second point, ice cold. So your solution does need to be really, really cold because that reduces any enzyme activity and prevents, for example, enzymes digesting your organelles. And then pH controlled or buffered. So we need to buffer our solution to make sure the pH is constant and at the optimum pH for the cells and the organelles. And that prevents denaturation of any proteins or enzymes present. The final part of this question asks you to explain how the student would isolate chloroplasts using a centrifuge. And the order is quite important in this question. So you need to be writing first about centrifuging at low speed to remove larger or denser organelles. And the classic one is your nuclei, because that's the largest, most dense organelle within a cell. And when you use a centrifuge, you put your solution into a centrifuge tube and the machine spins it fast, but you can change the speed. And if we start at a relatively low speed, once you've done the centrifuging, what you end up with is a pellet at the bottom of your tube, and at the top is a liquid supernatant. Okay? And if you spin at low speed to start with, what you'll get is in that pellet, you'll obtain your nuclei, so your larger, denser organelles, which will be your nuclei. And in the supernatant, you get any smaller, less dense organelles. What you would then do is extract your supernatant, leave your pellet behind, and then re-spin your supernatant at a higher speed. And what you'll find is if you re-spin at a higher speed, you can then extract your chloroplasts out in the pellet, and then any smaller organelles, such as lysosomes or ribosomes, would then be found in your supernatant. And you can use different speeds, and you can look these up, use specific different speeds to obtain different organelles. You don't have to learn any particular speeds, you just have to have an understanding of the relative size of organelles. So know that if you span at low speed initially, you'd extract out your largest organelles, which would be your nuclei. Slightly higher speed, you get things like your chloroplasts and your mitochondria. Even higher, you get things like lysosomes, and even higher than that, you get things like your ribosomes. Okay, so generally just increase your speed, and that means you get increasingly smaller organelles extracted out in your pellet at the bottom of your centrifuge tube. So that's the end of this video, in which we've covered the most tricky styles of exam questions within the cells topic. In the next series of videos, we will be looking at the cell cycle and mitosis.